Thank you, Dr. Kulkarni. Welcome, friends, colleagues, and industry partners. My name is John Buozis, and I'm from the School of Engineering, and I'll be talking to you about a biosensor for avian influenza virus detection using nanotechnology. So I'm going to start you off with a case study. So avian influenza A H5N2 was found in British Columbia in 2014. On November 27th, 2014, 28,000 birds became sick with the illness, but they didn't actually know it was that virus until about 4 or 5 days later. And the samples were sent out on the date, November 27th, where they discovered the symptoms. And the birds weren't destroyed until a further 5 other days. And during that time, due to this poor diagnostic turnaround, the additional farms were affected. 11 additional farms were affected between that time period. So a total of 240,000 birds died during this outbreak. And H5 and H7 subtypes are highly pathogenic and possess zoonotic transmission potential to humans. So due to this, governments and farmers alike are under immense pressure to ensure the health of both poultry and its consumers. So current detection methods. So the current detection methods are very invasive. It's invasive tracheal swabbing. So all of us, when we get sick, we go to the doctor. We have to get a throat swab if our throat is sore. It's very uncomfortable. You can just imagine how the chicken feels, right? The chicken's being held, being throat swabbed. It's not fun. Then the swabs are put in a media to preserve it, and then they're sent to the National Animal Health Laboratory or another facility that's equipped to deal with it. And there's shipping time, there's cost for the analysis, the man hours for the technicians, the reagents, and also just the cost of the preservatives as well. So we're trying to reduce that down. And you can imagine it would take a long time for the test results to come back if you're in a very remote farm. So you have to ship it to a city center, takes a few days. So, and at the lab, they perform polymerase chain reaction, so reverse transcription DNA amplification to determine the subtype and pathotype, and also they use sequence analysis for verification to see if it's H5 or H7. So there's a current technology gap because there's no pen-side or coop-side testing method right now. So also the virus has time to dangerously mutate during that transportation time and time to results. And influenza is linked to 250,000 to 500,000 fatal cases in humans annually. And that's because all influenza subtypes are from waterfowl, right? So even the regular influenza, you get the flu here, it's all from waterfowl. And there's a genuine need for rapid predictive technology. And this is just a little bit, this is just a little bit of a schematic here of how the disease transmits to backyard farmers. So basically, you can pick up the chicken, get the virus. There's also when the chickens have droppings in the litter. Also, when they run through it, it can become airborne as dust if they flap their wings and whatnot. So it's very dangerous. So the solution is to move from reactive techniques to predictive technology. And what I'm suggesting is, for example, here you take a droplet of blood from the bird. It has the virus. And this here is a biosensor. So basically it's nanotechnology dropped onto an electrode, and the biorecognition element is the antibody specific to the virus in question. So we can't currently work with H5 and H7 subtypes as they're both provincially and federally regulated. So right now we're just working with the surface proteins, but we do hope to move to that eventually because that is really needed in the field. So basically what you do is you drop the blood onto this electrode and then you put it into this transducer. It's pretty much like a diabetes blood glucose testing machine. So you just put the strip in and it will tell you whether or not the chicken has the virus. And so the takeaway message is that this is a platform technology that can be used for farmers, not only to test for avian influenza, but it could be extended to other poultry-related viruses and bacterial pathogens. Rapid on-farm detection of H5 and H7 of highly pathogenic influenza viruses could be done in the future, which would be outbreak control. And it's a tool for farmers moving from reactive to predictive methods. And lastly, a strain level identification is possible in the future using multiplexing. So basically, if a farmer gets sick, they go to the doctor. The doctor can then use this device to see whether or not it's avian borne illness or human flu. And that's the takeaway message from my presentation. Thank you very much.